Hello friends, today we are going to discuss about chromosomal aberrations. What is chromosomal aberrations? These are also known as structural aberrations, and as the name suggests, it is the change in the structure of the chromosome which is visible, or visible changes in the structure of chromosome. And this change may be either in the number of genes or in the arrangement of genes that are present in the chromosome. Origin of chromosomal aberrations: this is due to the breakage in chromosome. Then these broken pieces undergoes rejoining, but there is irregularities in rejoining. Hence, these are not rejoined in the same way as it was present in the normal chromosome, but in the different manner, and this leads to change in the structure of the chromosome, which is known as chromosomal aberration. Now let us see the factors that are responsible for the chromosome breaks. These can be cosmic radiation, nutritional deficiencies, and environmental conditions. And these occurs in almost all animals, but their frequency of occurrence varies. So the frequency of this chromosome breaks depends upon several factors like age, availability of oxygen, temperature, and metabolic condition. These can also be induced artificially by radiations like X-rays, gamma rays, beta rays, chemical mutagenic agents, that is alkylating agents, insecticides, herbicides, fungicides, and base analogues, and by several viruses. Now let us see types of chromosomal aberration. There are 4 types of chromosomal aberration: deletion, duplication, inversion, and translocation. Let us see them one by one. First is deletion. It is the loss of a segment of a chromosome, and depending upon the region where this lost segment is located, it is of 2 types. Terminal. When the terminal segment of the chromosome is lost, these also have telomere. Hence these are rare because the chromosome in which the telomeric end or the telomeric region is lost becomes unstable. If we see here, this is the normal chromosome having the genes A, B, C, D, E, F, G, H. Now the breakage occurs at this region. Then this segment is lost. This segment is present at the end. Hence it is known as the terminal deletion. Second is interstitial. Loss of intercalary segment of a chromosome. That is the region which is present in between telomere and centromere. This is the normal chromosome. Breakage occurs at this point, and this region BCD is lost. This region is present in between the centromere and the telomere, that is, it is intercalary. Hence this type of the deletion is known as interstitial deletion. We know that during the meiosis, the pairing of the homologous chromosome takes place. When chromosome with deletion pairs with its normal non-deleted homologue, then what happens? If we talk about terminal deletion, we can see this in the figure. This is the normal chromosome which have genes A, B, C, D, E, F, G, H, and this is the chromosome with deletion. Here its terminal end is deleted. So till A to E, these pair normally, but F, G, H do not have its counterpart in the deleted homologue. Hence these remains unpaired. And if we talk about interstitial deletion, This is the normal chromosome having the genes A, B, C, D, E, F, G, H. And this is the chromosome with deletion. And this deletion is interstitial. Here the segment having B, C, D is deleted. Now when the pairing takes place, A pairs with A, E pairs with E, and so on. But in the normal chromosome, B, C, D region do not have its counterpart in the homologue in which it is deleted. Hence it remains unpaired and these loops out during the pairing of the homologous chromosome. Now let us see genetic effects of deletion. If deletion is small, then it does not cause any harmful effect, because in case of the small deletion, it does not cause any detectable morphological change in the chromosome. Hence, no harmful effect is seen. But if the deletion is of larger size, then it is detrimental for the organism, or it is harmful for the organism. Act as a recessive mutants. Deleted genes acts as the recessive mutants because these deleted genes are not able to produce their phenotypic effect, because these are not present, these are deleted. Hence these are not able to express themselves. This is same as the recessive genes are not able to express themselves in the presence of dominant genes. Hence, it acts as the recessive mutant. Shows pseudodominance. In case deleted genes counterpart are recessive gene and these are present in the normal chromosome, then in the absence of the deleted genes, they express themselves as if they are dominant genes. Hence, these shows pseudodominance. If in the homologous chromosome, capital A and small a gene is present, this capital A is dominant and small a is recessive. If capital A is deleted from this chromosome and this small a gene is present in the normal chromosome, so in the absence of this dominant gene, the recessive gene expresses itself and behaves as the dominant gene. This is known as the pseudodominance. Crossing over is completely absent in the region of deletion, because in the absence of the deleted genes, its counterparts that are present in the normal chromosome are present in the hemizygous state, and in this condition crossing over is not possible. These have the harmful or lethal effects in diploids and haploids but not in polyploids, because in the polyploids extra copies of the genes are present, but in diploids and haploids the extra copies are not present, and hence in this case the deletion is harmful or lethal. Now second type of the chromosomal aberration is duplication. It is the occurrence of a segment of chromosome in 2 or more copies per genome. Types of duplication. Tandem duplication. When the repeated segment lies next to the original sequence and genes are in the same sequence. So this is the original segment having the sequence DE. This is the duplicated segment which is present adjacent to the original one, and it is present in the same sequence DE, DE. This is known as tandem duplication. Reverse duplication. When the repeated segment lies adjacent to the original sequence but with the reverse gene sequence. This is the original one and this is the duplicated one. Here the sequence is DE and here it is ED. So it is the reverse of the original one. This is known as reverse duplication. Now the displaced duplication. Duplicated segment lies in some other part of the chromosome. These can lie either in the same arm or in the arm different from the one having the original sequence. So if we see here, this is the original sequence, and this is its duplicated sequence. It is not present adjacent to the original sequence but present in the same arm as the original one. And here this is the original sequence DE, and this is the duplicated sequence. It is present in the arm different from the arm in which the original sequence is present. This is known as displaced duplication. Genetic effects of duplication. Duplication do not produces as drastic or lethal effect as deletions, but sometimes it produces harmful phenotypic effect. Example, bar eye keratin in Drosophila and Down syndrome in human beings. We are going to discuss about inversion. So what is inversion? Sometimes two breaks occur in a chromosome, and this break is followed by the rotation of the segment by 180 degree before rejoining. Suppose this is a chromosome and two breaks are taking place, one at this point and one at this point. This is the segment. This then rotates by 180 degree and then rejoins. Now what happens? The segment has the sequence EDC. Earlier it was CDE, now it is EDC. So here there is no change in the number of the genes. Here also it is 3, here also it is 3, but sequence of the segment is changed. The sequence of the segment is inverted. This is known as inversion. Now the types of inversion. There are two types of the inversion, paracentric and pericentric. First we will see paracentric. When both the breaks into the chromosome occur on the same side of the centromere and inverted segment is without the centromere. So if we see in this figure, both the breaks are taking place on one side of the centromere, here in this case on the right side of the centromere, and the inverted segment does not include the centromere, so it is without the centromere. This is known as paracentric inversion. Second is pericentric inversion. Inverted segment contains the centromere, that is, it involves one break on either side of the centromere. If we see this figure, this is the centromere, one break is taking place on the left side of the centromere and the other on the right side of the centromere. So this segment which is inverted includes the centromere. This rotates by 180 degree and rejoins. Now the sequence is CBAQ instead of QABC, and it is with centromere. This is known as pericentric inversion. Now let us see how the pairing and crossing over takes place between the normal chromosome and the chromosome containing the inverted segment. The normal pairing is disrupted and abnormal cytological configurations are produced. At time of pairing, the normal chromosome forms loop-like configuration to maximize pairing of homologous regions. This is the normal chromosome and this is the chromosome having the inverted segment. This is the inverted segment, its sequence is DCB instead of BCD in the normal chromosome. So this normal chromosome, after the duplication, it forms a loop-like structure so that there is the maximum pairing between the homologous chromosome. The other chromosome, which has the inverted segment, twists in the inverted region, and this chromosome which has the inverted segment DCB instead of the BCD twists in such a way so that the maximum pairing takes place between the homologous chromosome. So after the pairing, the crossing over takes place between the homologous chromosome, and we have seen that there are two types of the inversion, paracentric and pericentric, and the product of the crossing over in case of the paracentric and pericentric inversion is different. So we will discuss them one by one. So now let us see the pairing and crossing over in paracentric inversion. This is the normal chromosome. this is the chromosome with the inverted segment. This is the inverted segment. It has the sequence DCB instead of BCD. Then the duplication of this chromosome takes place, and after that pairing and crossing over takes place. The normal chromosome loops out in order to maximize the pairing, and the chromosome containing the inverted segment twists at the region where the inverted segment is present to maximize the pairing. Normal chromosome has the sequence BCD, but in the inverted chromosome the segment has the sequence DCB. So it twists so that the B pairs with B, C pairs with C, and D pairs with D. And after that, this is the point at which the chiasmata is formed. That is the region of the crossing over, or the point of the crossing over. The crossing over takes place between this second and this fourth chromatid. So the crossover product will be A, B, then the cross goes this way, C, D, A. So this, it has one centromere at this point and one centromere at this point. To this centromere, this normal non-crossover chromatid is attached, and to this centromere, this non-crossover chromatid containing the inverted segment, which is the third chromatid, is attached. So this crossover product forms a dicentric bridge to which one normal non-crossover chromatid and the non-crossover chromatid containing the inverted segment is attached. This is known as the dicentric bridge. And the other crossover product is HGFEDCB. This cross will go this way, BEFGH, 2 4 So this, we will get this one. This is the second crossover product. But here it is without the centromere. So it is known as the acentric fragment. So consequently it gets lost, because without the centromere, these cannot align itself or move. Now what happens to this centromere? Spindle fibers are attached, and this also spindle fibers are attached. During the anaphase, the spindle fibers move towards the opposite direction. This is pulled towards this direction and this is pulled towards this direction, and due to the two pulling forces in the opposite direction, in this dicentric bridge, random breaks occur. So during the segregation at the anaphase 1, this dicentric bridge is broken, and what we get? One of the fragment of the dicentric bridge and to its centromere, non-crossover normal chromosome is attached, and we get another fragment of the dicentric bridge, and to its centromere, non-crossover chromosome containing the inverted segment is attached. This is the inverted segment. So after the segregation during the second meiotic division, we get one normal chromosome, two chromatids which do not consist of all the gene sequences. So these are known as the deletion products, and one chromosome with the inverted segment, which is known as the inversion product, and it consists of all the genes. So what we can say, that products of crossing over in the paracentric inversion, one chiasmata in the region of inversion results. That is, during the crossing over, what we get? The formation of an acentric chromatid lacking centromere and a dicentric chromatid connecting the two chromosomes, which is known as the dicentric bridge. So as a result of crossing over, we get the acentric fragment, which is not viable, and we get the dicentric bridge to which two non-crossover chromatids are attached. Therefore, at the end of the meiosis, products are two non-crossover chromatids, one normal chromosome, and one inversion product, one acentric chromatid, two deletion products. This acentric chromatid and deletion product are inviable. So at last, what we get? One normal chromosome and one inversion product, which consists of all the gene sequences. These are viable. These we get the deletion products, which are non-viable. This acentric fragment is also non-viable. Now let us see pairing and crossing over in pericentric inversion. This is the normal chromosome, and this is the chromosome which has the inverted segment. The normal sequence of the segment is B, C, D, E, and inverted segment has the sequence E, D, C, B, and the inverted segment also includes the centromere. So it is known as pericentric inversion. Then the duplication of the chromosome and the homologous pairing takes place. So during the pairing, this normal chromosome loops out to maximize the pairing, and this chromosome having the inverted sequence or segment, these twists in the region where the inverted segment is present, so that B pairs with B, C pairs with C, D pairs with D, and E pairs with E, and this maximizes the homologous pairing. Then the crossing over takes place. This chiasmata is formed. This is the point of crossing over. What is the product of crossing over after the segregation? So crossing over takes place between the chromatids second and fourth. We get one of the crossover products A, B, C, D. Then this cross goes this way, E, A. A, B, C, D, E, A. This is one of the crossover product. So what is the second crossover product? G, F, E. This cross will go this way, D, C, B, F, G. So G, F, E, D, C, B, F, G. First and third are the non-crossover product. This is A, B, C, D, E, F, G. A, B, C, D, E, F, G, which is normal chromosome. And second non-crossover product is A, E, D, C, B, F, G. So this is also the non-crossover product. It has the inverted segment E, D, C, B. So what are the product of the crossing over? Two non-crossover chromatids, one normal and one inversion chromosome. What we get? Two non-crossover chromatids. One is normal and the other is inversion chromosome. Two chromosomes which have duplication for certain genes and deficiency for the others. So these are the crossover product. These have some of the genes in the duplicated form and there is deficiency of the some genes. If we see here A, B, C, D, E, A. Here A is present in the duplication form. B, C, D, E is present but F and G is not present. If we see here G, F, E, D, C, B, F, G. G and F is present in the duplicated form. E, D, C, B is present but A is not present. And due to the deficiency of the genes, these are rarely viable. Now, what are the consequences of inversion? Effect of inversion are not so drastic as deletion and duplication, because due to the inversion, number of the genes remains same. Only the sequence changes. Inversion affect phenotype because of position effect. What does this mean? If a gene is present at certain location of the chromosome, then it will show different action or express different phenotype. And if the position changes, it will not show the same action or express the same phenotype. So with the change in the position, the phenotypic expression or the action of the gene changes. This is known as position effect. And because of this inversion, the position of the gene changes and hence inversion affect the phenotype of the gene. Inversion lower the recombination frequency within the inverted sequence. Hence, inversion tend to retain the original combination of genes. Now, the significance of inversion. Inversion helps in the origin of new species. It also provides the proof for the occurrence of crossing over and also supports the view that only 2 of 4 chromatids undergo crossing over. We are going to discuss about 4th type of chromosomal aberration, that is translocation. What is translocation? It is the breakage of a piece of chromosome and its attachment to another chromosome. From one chromosome, a segment or piece of chromosome breaks and then it attaches to other chromosome. This is known as translocation. Now the types of translocation. First is non-reciprocal translocation. Transfer of a segment in one direction. Here this is one chromosome and this is the other chromosome. From the first chromosome a segment breaks. Breakage takes place at 2 points and this segment is then transferred and it attaches to the second chromosome. Here the break takes place at one place and then this segment attaches to it. Now this chromosome do not have that segment and this chromosome has the segment that breaks from this chromosome. So here the transfer of the segment takes place in only one direction. This is known as non-reciprocal translocation. Now the reciprocal translocation, this is the second type of translocation which is most common. Here a segment from one chromosome is exchanged with a segment from another non-homologous chromosome. Here from chromosome 1 a segment breaks and it attaches to chromosome 2. From chromosome 2 also a segment breaks and it attaches to chromosome 1. So the breakage and the attachment of the segment takes place in both the direction, or the transfer of the segment takes place in both the direction. These are the translocated chromosome. This chromosome has a part of this chromosome and this chromosome has a part of this chromosome. This is known as reciprocal translocation. Now let us discuss in detail how the pairing of chromosome in a reciprocal translocation takes place. These two are the normal chromosome N1 and N2 and these are the translocated chromosome T1, T2. From this chromosome, segment breaks and it attaches to this chromosome, and from this chromosome a segment breaks and it attaches to this chromosome. In this way it gives two translocated chromosome T1 and T2. So here there are two normal chromosome N1 and N2 and two translocated chromosome T1 and T2. Now the pairing of the chromosome takes place during the packaging stage of the meiosis 1. We know that homologous chromosome pairs during the packaging stage of the meiosis 1, but here the pairing is different. Let us say it. So during the packaging stage, in order to properly align translocated and non-translocated portion, criss-cross configuration is formed. This is the normal chromosome N1. A part of it pairs with translocated chromosome T1 and other part ABC pairs with the another translocated chromosome T2. This is the another normal chromosome N2. Here this portion pairs with the translocated chromosome T1 and this portion pairs with translocated chromosome T2. So in order to properly align the centromeres and the chromosome segments, criss-cross configuration is formed. So here we can see the pairing takes place among each other and all the 4 chromosomes then forms a criss-cross like configuration. Then what happens during the metaphase stage when the chromosome align along the metaphase plate? So here this is the metaphase plate and these are the 2 poles. These 4 chromosomes forms a ring or the circle-like structure, and these ring may be either twisted or these may be open. This is known as the alternate arrangement. Here this is the normal chromosome N1. This is T1, translocated chromosome. Normal chromosome N2 and translocated chromosome T2. And when these form the open ring-like structure, 2 types of the arrangement takes place, adjacent 1 and adjacent 2. In the adjacent 1, the arrangement is N1, then T2, N2, T1. In the adjacent 2, T2, N2, T1 and N1. Then the segregation. spindle fibers attaches with the centromeres and the segregation takes place during the meiosis 1. So how this segregation takes place? So here this segregation is two types, alternate and adjacent. In adjacent, adjacent 1 and adjacent 2. In the alternate, two types of the products or gametes are produced. One product has two untranslocated or the normal chromosome N1 and N2, and the another with the translocated chromosome T2 and T1. Here if we see the product which has the normal chromosome, then it has all the gene loci, hence it is viable. If we see this is known as the balanced translocation. This also has all the loci. Here ABC, this is balanced by DEF, similarly GHI and JKL. Here also it consists of all the loci, hence this is also viable or functional, and the offsprings that are produced by the fusion of the gametes are viable. Now in adjacent segregation, in adjacent one gamete consists of N1 T2 and the second consists of N2 T1. In the adjacent 2, one gamete has N2 T2 and the other has N1 T1. And all the products or all the gametes have duplication and deletion product. If we see here, so the N1 has A, B, C, D, E, F, T2 has A, B, C, J, K, L. Here ABC is in duplicated form. GHI is not present, and JKL is present. Similarly here GHI, JKL is present. GHI is present in duplicated form, DEF is present and ABC is absent. Similar is the case here and here. So here duplication and deletion products are produced, and these are inviable or less viable, or we can say non-functional. So the offspring that we get are less fertile or less viable. Robertsonian translocation. It is a type of reciprocal translocation. We have already discussed reciprocal translocation in the translocation video. You can refer that video to understand it better. This Robertsonian translocation was discovered by American biologist William Rees Robertson in grasshopper in 1916, hence the name Robertsonian translocation. It is also known as whole arm translocation or centric fusion. It occurs in acrocentric chromosome. In humans, chromosome number 13, 14, 15, 21 and 22 are acrocentric chromosome. Now what is acrocentric chromosome? Acrocentric chromosome has centromeres situated such that one chromosomal arm is much shorter than the other arm. This is the acrocentric chromosome. Here centromere is situated near one end. So one arm is very short and the other is long. Now what is Robertsonian translocation? It is a chromosomal abnormality where two chromosomes break. These two chromosomes are acrocentric and the long arm fuse to form a single chromosome with single centromere. Short arm also join to form a smaller reciprocal product and is usually lost within a few cell division. But this loss of the smaller reciprocal product does not have any ill effect to the individual, because these short arms consist of non-essential genes which is also present elsewhere in the genome. So let us understand this. These are the two non-homologous acrocentric chromosomes. This chromosome breaks at this point and this chromosome breaks at this point. This long arm and this long arm with the centromere fuses. Now it behaves as a single chromosome with centromere. These two short fragments also joins, but this is gradually lost, or consequently this fragment is lost. So here this fragment of acrocentric chromosome is exchanged for this fragment, and this fragment of the chromosome is exchanged for this fragment. So it is an example of reciprocal translocation. Now let us see the effect of Robertsonian translocation. What happens when Robertsonian translocation takes place in any individual? When in Robertsonian translocation, long arm of one chromosome joins with the long arm of another acrocentric chromosome, as seen in the figure. The heterozygous carrier is phenotypically normal. So the carrier in which this translocation takes place is normal. But the offsprings that these carriers produce may inherit an unbalanced trisomy. Trisomy means that these have extra copy of any chromosome, and this causes certain syndromes. In some cases, certain syndromes are seen in the offsprings. Example, trisomy 21, Down syndrome, where extra copy of chromosome number 21 is present. Trisomy 13, Patau syndrome. In this, extra copy of chromosome number 13 is present. This type of the Robertsonian translocation occurs in 1 in 1,000 newborns, and most frequently it occurs between these acrocentric chromosomes 13 and 14, 14 and 21, and 14 and 15. Now let us understand the effect of Robertsonian translocation with the help of the diagram, or let us discuss how the Robertsonian translocation affects the progeny. So this is the individual, it is 2n, total number of chromosome is 46. We are showing chromosome number 14 and 21 because here we will explain how Robertsonian translocation between the chromosome number 14 and 21 affect the progeny of this individual. So there will be 6 possible type of the gametes that this individual can produce during the gametogenesis. This is the normal gamete with one copy of each chromosome number 41 and 21. This gamete has 14 by 21, chromosome number 14 and 21, which shows the Robertsonian translocation. Here one copy of the normal 14 chromosome is present plus 14 by 21. Here one copy of 21 chromosome is present. Chromosome number 14 is missing. Here one copy of chromosome number 14 is present. Chromosome number 21 is missing. Here if you see, one copy of the normal chromosome number 21 is present and one 14 by 21 chromosome is present. Now when these gametes are fertilized by the normal gamete containing one copy of chromosome number 14 and one copy of chromosome number 21, then the zygotes or the offsprings that we get, if this gamete fuses with this gamete, so both are normal, the individual that we will get will be normal, containing 2 copies of chromosome number 14, 2 copies of chromosome number 21. If this fuses with this gamete, so it will have one copy of normal chromosome 14, one copy of normal chromosome 21 and one 14 by 21. So this individual will be balanced carrier because these have genes present in the chromosome number 14 and genes present in the chromosome number 21 in 2 copies. So it will behave normal, but it will be carrier. It can affect its progeny if this normal gamete fuses with this gamete. So here there will be 2 copies of the normal chromosome number 14, one copy of normal chromosome number 21 and one chromosome 14 by 21 showing the Robertsonian translocation, as a result of which there will be 3 copies of chromosome number 14 and 2 copies of chromosome number 21. That means there is one extra copy of chromosome number 14. So the individual will have 47 chromosome instead of 46 chromosome with one copy of extra chromosome 14. So it is known as trisomy 14. If this gamete fuses with this gamete, then there will be 2 copies of chromosome number 21 but only 1 copy of chromosome number 14. 1 copy of chromosome number 14 is missing. So the total number of chromosome will be 45 and this is known as monosomy 14 if this normal gamete fuses with this gamete. So there will be 2 copies of chromosome number 14 but only 1 copy of chromosome number 21. Again this will have total number of chromosome 45 instead of 46 chromosomes because 1 chromosome 21 is missing. So it is known as monosomy 21. If this normal gamete fuses with this gamete, so there will be 1 copy of the normal chromosome 14, 2 copies of the normal chromosome 21 and 1 copy of chromosome 14 by 21. Thus there will be 2 copies of the chromosome 14, 3 copies of chromosome 21. That means 1 extra copy of chromosome 21 is present. So there will be total 47 chromosome. This is known as trisomy 21 because it has extra copy of chromosome 21 and this leads to Down syndrome in individual. Down syndrome we have already discussed under the topic aneuploidy. You can refer my video, the link is given in the description box. This is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.